Good afternoon, student. My name is Assistant Professor Vaishali Sonawane from Department of Microbiology. I am presently working in MVP's S.V.K.T. Arts, Science and Commerce College, Deolali camp. Today's lecture, topic for today's lecture is bacterial surface structures. So, before beginning with our lecture, we'll have a brief recap of what we have discussed in our previous lecture. So, this particular lecture, bacterial cell surface structures, are meant for FYBSc microbiology student. So, let's see the recap of what we have discussed in our previous lecture. So, in last lecture, we have discussed about the cell wall of gram-positive and gram-negative bacteria. Along with that, we have also gone through the structure of cell membrane. Under that, we have discussed about the fluid mosaic model of the cell membrane. All these aspects we have discussed in concern with the bacterial cell. So, today's session agenda, what we are going to discuss in our today's lecture. Firstly, we are going to focus on the bacterial surface structures. So, various structures that are present on the surface of bacteria that we are going to see. Under that, we are going to see capsule, slimy layer, flagella, fimbriae and pili. So, these are the various surface structures which we are going to see in our today's session. So, let's begin with today's lecture. When we talk about surface structure of a bacteria or surface appendages, these are the structures which are present on the surface of the bacterial cell as the term indicates. So, some bacteria have a what you can say layer, slimy layer or layer which surrounds the surface of that particular bacterial cell. So, all these surface structure includes all the all the structures which are mentioned here. It includes capsule, slimy layer or your flagella, pili, fimbriae, all these are considered as the surface structure of bacteria. And when we talk about all these surface structures of bacteria, it is not necessary that each and every bacteria is going to have each and every surface structure which is mentioned in this particular. Some bacteria may have one or two, or some may have more than 2 to 3 different types of surface structure on their surface. So, let's begin with the capsule. So, capsule is nothing but it is polysaccharide in nature and it covers the or you can say it is the outermost part of the cell. If the cell is capsulated, it envelopes the cell completely. It is well organized and tightly packed, and therefore it makes the cell resistant to the staining procedure. When we talk about capsule, it is near about 0.2 micrometer in thickness and it is quite viscous, which is firmly attached to the cell. If capsule is not that much thick, if the thickness of capsule is less, then in that case it is called as slime or it is called as your slimy layer. And when we talk about slimy layer, it is quite loosely attached to the bacterial cell. But when we talk about capsule, it is firmly attached and it is not easy to remove the capsule so easily from the cell, whereas your slimy layer can be easily removed from the cell. If we see the composition of capsule, near about 98% of the capsule content is the water and 2% is the polysaccharide. So, basically it is ample amount of water is present, and in case of some the polysaccharide component or the sugar which is present in the capsule can vary from organism to organism. Like for in case of acetic acid bacteria, it contains homopolysaccharide. Like in case of leuconostoc, it contains cellulose or it contains a glucose or fructose as a sugar. Or in case of Klebsiella pneumoniae, if we see in the case of Klebsiella pneumoniae, it is made up of glucose, galactose or ribose. These are the sugars which are present in the capsule of these organisms. Similarly, Bacillus anthracis, if we see the capsule of Bacillus anthracis, it is made up of polypeptide which contains polymer of D-glutamic acid, or you can say glutamate. when we talk about capsule, it is a delicate structure, even though it is firmly attached, but it is a quite delicate structure. And if vigorous washing is given to the cell which is having a capsule, in that case capsule can be removed, or your organism, or even the process of subculturing, it result in loss of capsule from the cell. And when we talk about capsule, it is very important part, or it plays very important role as a virulence factor. This thing we are going to discuss when we are going to see the functions of capsules. And when we talk about staining of capsule, so for staining of capsule we generally go for negative staining. So negative staining is the one in which we are not going to stain the capsule. Instead of that, we are going to stain the background as well as the bacterial cell, and your capsule will appear colorless in contrast to the color of background and the bacterial cell. So that is called as your negative staining. So this is the image which shows the capsule and a slimy layer. Okay, so you can see the capsule is very firmly attached, whereas your slimy layer is loosely attached to the bacterial cell, and it is quite, capsule is quite uniform, whereas your slimy layer is not uniform. These are some images of stainings of your capsule. So you can see the colorless zone which is appearing, that is the capsule, whereas there is pink color microorganism, bacteria, and the background is colored. So in contrast to the colored bacteria and background, capsule appears colorless in nature. So these are some of the images of staining procedure or negative staining that is basically employed for your capsule. Now there are two types of capsules which is present in case of bacterial cell. One is macrocapsule and another one is your microcapsule. Macrocapsule is the capsule which has a thickness of 0.2 micrometer, or it is more. It is quite thick and you can easily observe it under microscope. You can observe that capsule under microscope by performing negative staining. But another type is your microcapsule. Microcapsule, as the name suggests, it is the capsule whose thickness is less than 0.2 micrometer, and for visualization of that type of capsule you require an electron microscopy. So a microcapsule is not easily visible to under a light microscope. Moving on to the functions of capsule, there are various functions which are played by capsule. So we'll have a look on all these functions one by one. The very first function is the prevention of desiccation and drying of cell. So since in the composition itself we have seen that capsule is made up of 98% of water. So that 98% or that huge amount of water protects the cell from dry condition and desiccation, and therefore it enhances the survival of that particular organism. It protects the capsule, it protects the bacterial cell from the injury like temperature, drying, or it also protects from the osmotic shock. Then the very important thing is the attachment. Generally what happens when we talk about pathogenic microorganism, now when we talk about pathogenic organism, to establish an infection it is very necessary that the organism should attach to the host cell, and a capsule is the one which generally helps in attachment of pathogenic microorganism to the surface of the host cell, and therefore helps in the establishment of microorganism or establishment of pathogenic organism, and along with that it also leads to the disease. It helps in establishment of organism along with the symptoms and disease, occurrence of disease. For example, the example here is given Streptococcus mutans, which causes dental infection or dental caries attachment of these organisms to the teeth. Capsule enhances the attachment of this organism to the tooth. Another character is that it is antiphagocytic in nature. Antiphagocytic in nature means that presence of capsule helps the organism to escape the process of phagocytosis. We all know that our body has 3 lines of defense, and as a part of second line of defense, when any foreign organism or foreign material or antigen enters inside the body of host, your body, the macrophagic cells or the phagocytic cell, they try to engulf it to clear the organism. But presence of capsule over the surface of the cell helps that organism to escape the process of phagocytosis. Therefore, it also acts as a antiphagocytic in nature. Then capsule, along with that, capsule also make the organism resistant to bacteriophage. So bacteriophages are the phages which infects the bacteria. So it makes that organism resistant or protects the organism against bacteriophage infection. Along with that, it also serves as a source of nutrition for the organism. Since it contains polysaccharide, it contains sugar, and along with that it contains water. So it provides nutrient to the cell. Repulsion is another function that is played by your capsule. Some charged capsulated bacteria, they repel from each other, and this also helps in avoiding the competition between the bacteria for the sake of space or for food. So let's see some of the examples of the microorganism or the bacteria which contains capsule. So the example is Bacillus ceteris, Bacillus anthracis. So generally genus of Bacillus contains the capsulated one. Then Streptococcus pneumoniae, it is the well-known organism which causes pneumonia, Klebsiella pneumoniae, then Haemophilus influenza, then Clostridium perfringens, Neisseria meningitidis. These are the various organisms which harbors the capsule, and all of them are generally pathogenic in nature. So pathogenicity, you can say, is directly related to the presence of capsule in some case. Like for one good example is Streptococcus pneumoniae. So loss of capsule from the Streptococcus pneumoniae, it makes that organism non-pathogenic. So if capsule is removed, that in that case organism is not able to cause the infection. Then the next surface structure is the flagellum. So flagella are basically known as the locomotory organs, locomotory structures in prokaryotes. So if an organism is said to be motile, okay, if an organism, we are saying if the organism is motile, so in that case that organism contains the surface structure known as flagella. So you can say that flagella is the locomotory organ which is, or locomotory structure which is present in some microorganisms. So if we talk about the structure of flagella, it is long, thin, near about 20 nanometer in length, and whip-like appendages is there. It is attached to one end and it is free to another end. Now, when we talk about flagella, like capsule, we have just seen the capsule. So, you can easily observe the capsule under light microscope. But when we talk about flagella, they are so thin that you are not able to visualize them under light microscope. And if you want to visualize them or if you want to observe them, you need to go for electron microscopy, and a special staining, flagellar staining is there, that can be performed to visualize the flagella of the bacteria. Now when we talk about flagella, generally most of the rod shaped bacteria, contains flagella, whereas most of the cocci are non-flagellated ones. Means you can say 99% of rods are flagellated and they are motile, and 99% of cocci are non-flagellated and are non-motile, but exceptions are always there. Those bacteria which lacks the flagella are called as atrichous. So, atrichous organisms are the one which doesn't have a flagella. So let's see the structure of flagella. So flagella is basically proteinic in nature. It is made up of the protein known as flagellin, and it contains, it is made up of different structures. So it is made up of what you can say basal body, filament and a hook. So hook is the connecting link between basal body and the filament. Filament is actually your flagella. So the basal body, as I told you, since it is the basal body, it is attached to the cell membrane or the cell wall of the bacteria. Hook is the one which binds, which connects your filament with that of your basal body, and it contains, your basal body generally contains rings, which along with via that rings your filament passes. In gram negative, two sets of rings are present, whereas in gram positive it lacks lipopolysaccharide layer. Therefore your L ring, right, L ring is absent. This we are going to see in our next slide. You can see. So gram positive organism have basal body and it has two ring, that is P and M ring is there. Gram negative are the one which has 4 because they have an additional lipopolysaccharide layer. So these are the 4 rings which is present in gram negative. L ring, it is associated with lipopolysaccharide layer. P ring, it is associated with peptidoglycan layer. M is the one which is associated with plasma membrane, and S is the one which is attached to the inner part of your plasma membrane. So these are the 4 rings, LP, SM. These are the 4 rings which are present in gram negative, and the only 2 rings are present in your gram positive bacteria. So this is, you can see, this is the structure of your flagella. You can see the 4 rings. All these 4 rings comprises the basal body of the flagella. L ring, P ring, followed by S ring, and last is your M ring. Outer membrane, peptidoglycan, cytoplasmic membrane, all these components, via all these layers your ring passes. And along with that you can see the hook. Hook is the one which binds your basal body with the filament, and filament is the one end opened at one end. So this is an image which shows the different types of flagellar arrangement in a bacterial cell. Now when we talk about presence of flagella, it is not necessary that each and every bacteria is going to have flagella all over the surface of their body. But accordingly, there are different types of flagellar arrangement. So we are going to see one by one. The very first type of flagellar arrangement is the Monotrax. Monotrax is, from the name itself, you can identify that mono is the single and trachus is the your flagella or a moving organelle. So Monotrax is the arrangement in which there is presence of a single flagellum at one polar end. So your bacteria is going to have a single flagella at one end. So that type of flagellar arrangement is said to be Monotrax. Second one is Lophotrax. Lophotrax is the type of flagellar arrangement in which a bunch of flagella are present at one end. So more than one flagella is present, but they are restricted to one polar end. So that type of arrangement is called as your Lophotrax. Third one is the Amphitrax. In Amphitrax arrangement of flagella, there is presence of a single flagella at both the end, means at both polar end there is presence of one flagella. So that type of arrangement is called as your Amphitrax. Then next is the Peritrax. Peritrax flagellar arrangement is the arrangement in which flagella are distributed throughout the surface of the cell. So your whole cell is covered with a flagella. So one good example of Peritrax flagella, organism having Peritrax flagella, is your E. coli. And Amphilophotrachus. Amphilophotrachus is the arrangement in which there is presence of bunch of flagella, but these two bunches are present at both polar end. Okay, and one is Ertrachus. Ertrachus, we have seen that the organism without your flagella. So these are the various types of flagellar arrangement which is present in a bacterial cell. Now let's see what are the functions of flagella. So very first thing is it is a primary organ, primary structure for locomotion. So presence of flagella makes that organism to move from one place to another. It also help in colonization of the microorganism, and along with that it also help in spreading of infection, spreading of infection from one side to another side. Means what? When a organism is flagellated one, so if it is causing infection at a particular site, so if the organism is non-motile, in that case your infection is going to restrict at that particular site only. But if the organism is flagellated one, the organism can move from one site to another to establish a new infection. So presence of flagella is also one of the virulence factor that is associated with your bacterial cell. And in case of non-pathogenic organism, this we talked about pathogenic, but in case of non-pathogenic organisms which generally resides in soil, in that case presence of flagella help in colonization of that particular organism. Along with that, in some bacteria, flagella are involved in exchange of nutrients and waste material. In alkalophilic bacteria, they are flagella are sodium driven pump which help in maintaining the osmotic balance, along with that concentration of sodium in the cytoplasm. So these are the various functions of your flagella. So let's see next organelle, that is fimbriae and pillae. So you can see in this particular image, it includes all three organelles. Your flagella is there, pillae is there, and fimbriae is there. So fimbriae are they are large, many in number as compared to that of your flagella. So and they are spread throughout the cell surface. They are the smallest you can say appendage, surface appendage which is present in on the surface of bacterial cell. So when we talk about pillae, you can see in this particular image another similar thread-like structure which you can see is the pillae. Now these pillae are also called as your sex pillae. So fimbriae and pillae, basically they play important role in the attachment of the bacterial cell. When we talk about fimbriae, fimbriae plays very important role in the attachment of mucosal cell lining in case of pathogenic microorganism. But when we talk about non-pathogenic organism or when we talk about pillae, pillae are the one which basically are involved in your genetic exchange or sexual reproduction in case of your bacteria. So let's see the basic difference between fimbriae and pillae. So this is the table which is showing a basic difference between fimbriae and pillae. So in the first column the various characters are mentioned and the basic difference between fimbriae and pillae is given. So if we talk about fimbriae, fimbriae are tiny bristles like fibers which arise from the surface of bacterial cell. Whereas if we talk about pillae, pillae are hair-like microfibers that are thick tubular structure and it is made up of pillin. So the protein from which your pillae is made is called as pillin, whereas fimbriae is the one which plays important role in the attachment of your cell. If we talk about lengthwise, so fimbriae are shorter than pillae, whereas pillae are quite longer than fimbriae. Diameter, if we talk about fimbriae, they are thin in diameter, whereas pillae are thicker than that of your fimbriae. Number of the pillae which is distributed throughout the bacterial cell, you can see number of fimbriae is 200 to 400 per cell. But when we talk about pili, they are less as compared to that of your fimbriae. It ranges from 1 to 10 per cell. Fimbriae is made up of protein known as fibrillin protein, and pili is made up of protein called as pillin protein. Rigidity wise, the fimbriae are less rigid, whereas pili are more rigid than that of your fimbriae. And fimbriae are found both in gram positive and gram negative organism. But when we talk about pili, they are generally associated with gram negative bacteria. If we talk about the formation, how these things are present, or you can say formed in a cell. So, formation of fimbriae is governed by the bacterial gene. So, bacterial genome itself is going to decide whether fimbriae is to be formed or not. But when we talk about pili, pili is not regulated by the bacterial genes on the bacterial chromosome, but it is governed by the plasmid gene. So, if a plasmid is there, and that is generally are plasmid. If plasmid is there, so that plasmid, if that plasmid is going to have a gene which is going to code for pili, only in that case it is going to have pili. Then if we talk about the function, so responsible for, if we talk about fimbriae, it is responsible for surface to surface, cell to surface attachment, and it enables the cell to adhere to the surface and other bacteria. But when we talk about pili, it is, as I told you, it is responsible for sexual reproduction, which is basically the conjugation, and gene transfer. This is the important function which is played by your pili, and along with that it also helps in attachment of pathogenic organism. Next is the motility. They generally pili, if we talk about pili and fimbriae, as compared to these two, your flagella is the one which is the organ of your locomotion in case of bacteria, and fimbriae does not participate, or it does not impart the motility to bacterial cell. But when we talk about pili, there are type 4 type of pili, which type 4 pili which is associated with a special type of motility, that is called as your twitching motility. There are no receptors for others in case of fimbriae, whereas pili can serve as a receptor for certain viruses. So, attachment of that viruses on the surface of that bacterial cell. So, if we talk about lastly, we will see the examples. So, Salmonella typhimurium, Shigella, species of Shigella like Shigella dysentery is there, or most of the bacteria which infects your gastrointestinal tract, they generally have a fimbriae which provides that, which helps that organism to attach to the mucosal cell lining on which is present inside the intestine. And because what happens ki when, if an organism has to establish an infection, the very first thing is that the organism should establish himself into the body of host, and it should produce toxin, or it should increase in number to attain the minimum dose which can cause the infection. So, in case of intestinal infection, the organism produces toxin and can cause diarrhea. In case of pili, it is present in Escherichia coli, it is present in Neisseria gonorrhea. In case of Neisseria, it is basically cause of gonococcal infection, or gonorrhea, which is infection that is associated with urogenital and cervical epithelium. So, these are the various functions and the basic difference between pili and flagella. So, let us see summary what we have discussed in our today's lecture. So, in today's lecture we have discussed all the bacterial surface appendages or structures. So, under that we have discussed about capsule, we have seen slimy layer, we have seen flagella, pili and fimbriae. All these surface structures generally contributes to the virulence of the pathogenic microorganism. So, on the basis of what we have studied in our today's lecture, there are two questions for your assignment. First is the diagrammatic representation of flagella in gram positive and gram negative bacteria, and second one is write the note on the flagellar arrangement, various type of arrangement of flagella that we have already seen in our today's lecture. So before moving on, we'll see a brief recap what we are going to see in our next lecture. So in next lecture we are going to see some other cell inclusion. Basically we are going to discuss the cell inclusions. So under that we are going to see ribosomes, and under cell inclusion we are going to see gas vesicle, carboxysomes, PHP granules, metachromatic granules, glycogen bodies, magnetosomes, sulfur granules. All these things we are going to see in our next lecture. So that's all with today's lecture. Thank you.